Hello everyone and welcome to Biology. I am Diler and today I'm going to talk about chromosome mapping and mutations. The farther apart two genes are located on a chromosome, the more likely crossover will occur, the greater the percentage of F2 offspring showing recombinant traits. The farther apart the genes for those traits must lie on a chromosome. Researchers conduct breeding experiments and use the resulting data to prepare a chromosome map. A chromosome map is a diagram that shows the linear order of genes on a chromosome. Alfred Sturtevant, one of Morgan's students, made the first chromosome map for flies, as shown in this figure. To prepare his map, Sturtevant compared the frequency of crossing over for several genes. The percentage of crossing over for two traits is proportional to the distance between them on a chromosome. Sturtevant defined one map unit as a frequency of crossing over of 1%. Okay, now let's get to mutations. Cystic fibrosis results from a mutation. Mutation is a change in the nucleotide base sequence of a gene or DNA molecule. Germ cell mutations occur in an organism's gametes. Germ cell mutations do not affect the organism itself, but they can be passed on to offspring. Somatic cell mutations take place in an organism's body cells and can therefore affect the organism. For example, certain types of human skin cancer and leukemia result from somatic cell mutations. Somatic cell mutations cannot be inherited. Lethal mutations cause death, often before birth. Some mutations, however, result in phenotypes that are beneficial to the individual. Organisms with beneficial mutations have a better chance of surviving and reproducing, and therefore have an evolutionary advantage. Mutations provide the variation upon which natural selection acts. Mutations can involve an entire chromosome or a single DNA nucleotide. Okay, now we have chromosome mutations. Chromosome mutations involve changes in the structure of a chromosome, the loss or gain of a chromosome. Three types of chromosome mutations are shown in this figure. A deletion is the loss of a piece of a chromosome due to breakage. In an inversion, a chromosomal segment breaks off, flips around backward, and reattaches. In a translocation, a piece of one chromosome breaks off and reattaches to a non-homologous chromosome. In non-disjunction, a chromosome fails to separate from its homologue during meiosis. One gamete receives an extra copy of a chromosome, and other gamete receives no copies. An example of non-disjunction that results in Down syndrome is shown in this figure. Okay, now let's get to gene mutations. The substitution, addition, or removal of a single nucleotide is a point mutation, which is a change that occurs within a single gene or other segment of DNA on a chromosome. In a substitution, one nucleotide replaces another, as shown in this figure. If this substitution occurs in a codon, the amino acid can be changed. In a deletion mutation, one or more nucleotides in a gene are lost. This loss can cause incorrect grouping of the remaining codons, called a frame mutation, making all amino acid downstream change. A frame mutation, as you can see in this figure, these mutations in turn can have a disastrous effect on the protein's function. In insertion mutations, one or more nucleotides are added to a gene, which can also result in a frameshift mutation. And here we have come to an end. Don't forget to subscribe our channel on YouTube and follow us on Instagram. Until next lesson, may God be with you guys.